out of oil, have mud volcanoes, and that mimics some of the environments we see in Titan when the Saturn moons. So you can understand kind of what kind of organisms live there and how could we try to find them elsewhere. So if you like to travel to really wild places, this is a good career choice potentially. So what do we kind of start to figure out about life? We haven't found, there's a question about that. We haven't found life elsewhere, okay? Just to make it clear, not yet, although the odds are that we will at some point, probably. But as you can see in this graph here, and I'll just point you to what is important that I'm trying to point out here, but this is the story of life on Earth, right? When it appeared, and Earth itself was formed, just a little 5.5 billion years here. So life emerged shortly after that. But you can see this great oxidation event here is like a peak. This is the release of oxygen, right? The release of oxygen into Earth's atmosphere. It wasn't here to begin with, but this is what we all breathe, right? None of us could be alive without oxygen. Now, what's interesting in this diagram, as you look at it, is that you see this word eukaryotes. That's a particular domain of life, as a form of life that is what we are, like what our cells are. But that's not what bacteria are. They're a different domain that we'll just talk about. But what you see is that the oxygen appeared before our types of cells, which has also the same cells as plants. And we all think of plants as being the ones that release oxygen into our atmosphere, right? Which they do these days and have maintained it such and with some peaks. But that big peak that came from 0 to right up here, this was bacteria alone in our planet. This is it's called cyanobacteria. And it's mind-blowing to me, and I think should be for most people, that something that you can't even see, which is microscopic, right? This bacteria you can't even see, have completely transformed the whole planet, right? They have put oxygen into the atmosphere, actually as a weapon against other microbes, because it's toxic to a lot of microbes they are killed by oxygen. They did this to kind of win those battles, but it's crazy to think what these small creatures can do to a planet, and then you can think about what they could do to your body, right? Which is actually much smaller in terms of scales, right? Now, another humbling thing. What is this? Does anybody know what this is? Yeah. Yeah, sure.
I think it's a tree of life, like universal. It's called the universal tree of life.
Yep.
I know that because my dad did these studies before.
Very good. Yeah. Phylogenetic tree, a tree of life. Yep. What does it represent? Do you know how to, or does anybody know how to read these things? No? Yeah.
All right. So there are different classes of animals in the phylogeny with like kingdoms that are like classified class that.
Right.
And then there's different like groups of animals, like different like branches of the evolutionary tree according to the theory of evolution.
Yeah, so somewhere along those lines. So one of the things to center folks is it says here the root, right? So the root, this means that we don't quite know where the origin of life is exactly, but according to the phylogenetic tree, and this is based on genes, right? So it's like you get all the genes of all the organisms we know, and we put them together and compare them, all right? And how you read the tree of life is really the further away things are from each other, the more different they are. The closer they are, the more alike they are, right? And so you see that we think the root is about here, and you can see these big names: bacteria, archaea, eukarya. These are what 3 domains of life have been identified as, because they're so distinct, you can see them, right? There's 3 groups here. Bacteria is what you think of as the microorganisms. Archaea are also microorganisms, although they're a little different. Their metabolism is different, and they tend to be in extreme environments. So hot springs, think salt flats, kind of very hydrothermal vents at the deep ocean. they are a little different. They don't tend to be associated with disease or anything like that. And then there's eukarya, which is us, as we were talking about, our cells. And what's funny here is that we know the root is about here, and we know we're here. This is Homo sapiens. This is where we are, right? And in this scale of things, of how different we are from each other, you can see what are the closest two things that we have to us there. Anybody read the names of the things we have there? Yeah. Yeah. Does anybody know what that is? The other things that aren't shown are so similar to us, it can't be shown because it's too close, right? So these are the two closest things to us. And I can tell you that the two closest things to us that can be shown without being exactly like us pretty much is zea, which is actually corn, and this is a mushroom, okay? So everything else you can think of, a mouse, a plant, or whatever, right? It's gonna be actually like us, genetically, right? In this comparison, right? So you can appreciate how different bacteria are from each other, right? The distance between each one of these microorganisms is extremely different compared to the difference that separates between us as corn and mushrooms, right? So it's really humbling to think about what bacteria can do, what they're able to produce, to release to the atmosphere, do all these things. This metabolic capacity is really very, very distinct from each other. Some of the things that we did in this work, was done over a few years, as you can imagine. But this is experiments that we do also in preparation to the mission to Mars, right? That is coming. But the Biomex Biology in Mars experiment, this was actually done in collaboration, not just NASA, but also the European Space Agency, or ESA, and actually Roscosmos as well. Russian Space Agency was as well involved, and Russian cosmonauts. What we did here was to really do the experiment of, what's going to happen to us when we go to Mars, because nobody actually knows very well, because we haven't really left the protection of Earth yet. So we don't know in terms of radiation, how we're going to do us and our microbes. And so what we did was to collect these microbes from very exotic places on Earth, and then put them in what's called a Martian simulator, which at the DLR, which is the German Aerospace Center. And they can simulate conditions, and then we saw which ones could even survive Martian conditions. And the ones that were selected were that survived that experiment were collected in Antarctica, so South Pole, a lot of them. And then we put them in chambers, sent them up to the International Space Station, and then we told the astronauts and cosmonauts that we really needed them to put this outside the shuttle, because we wanted to receive all that radiation, the cosmic radiation, in full force. So they did that in extravehicular activities. They placed them there, and then we said, just leave them there now. And we opened them and closed them for some experiments. And then after 2 years, we waited 2 years because the longest experiment was done this way. After 2 years, we told them, okay, bring them back down so we could take a look. So we got the samples. And you can see just the little samples here that we got back. You can appreciate how in 2 years they changed just by looking at it, right? There's this fluffy green stuff here. And then, after a while they changed. And then after the 2 years, they were really very different. And we have published these results in several papers, but it was, showing that these bacteria survived, but they changed a lot. And the things that changed are related to things like biofilm formation, for example, which can have a big impact in how they relate to us. We often forget about other things. We think about what happens to the human body as we go through space, but actually what happens to our microbes is just as important, because they're also being impacted by these conditions, and we can't just think that they're not going to change. They are going to change, quite massively, right? So, Mayo at the time approached me to join the Human Microbiome Program here. And this was actually the line they used to recruit me. It was, can you look for aliens in the human body? They were trying to put together the Human Microbiome Project. And how many of you know what the microbiome is? Anybody? You want to? Are they good or bad? Could be both. That's right. Can they be good and then become bad? The same one? They can. That's right. Just like you. You know, it's yeah. Microbes are complicated, and even more interesting is that sometimes if you put two microbes together and then you introduce a third, and these were friends, now they stop being friends. They do that too. So it's very interesting. Social life of microbes. But that was the pitch that was given to me, and I thought that was a really interesting thing. And I accepted under the condition that I could continue to look for aliens somewhere else too, and that was a mutual agreement, so that was great. So yeah, human microbiome. These are microbes that live in your body. I know all your, you know, especially in early, you know, pre-K and elementary, you know, your teachers tell you germs, you know, wash your hands, do this, do the other. You know, as you grow up and grow older, you know, you understand that there's actually some microbes that are very beneficial to your health. They're not all pathogens, they're not all bad, and some of them are actually essential for your health. So those we should try to protect. And this is just some numbers for you to think about, you know, what kind of impact they have on your body. There's about 10 trillion cells in our body, but there's about 100 trillion microbial cells. And whereas we have about 20,000 genes, there's about 20 million genes that are microbial in nature, right? So when you eat something, when you take a medication or whatever it is, that's a lot of cells and genes to act on that thing, right? That is beyond your direct control. They are doing that. And sometimes they do it before your body has a chance to do anything about it. So that's very important to think about in terms of health. If you have any doubts about this, you can do this experiment with your own hands, right? Just place your hand on a petri dish and you get your kind of bacterial fingerprint, right? This is actually being used by some places and investigators to use this for forensics, for crime investigation, that each one of us has a particular microbiome signature that can be used to do this sort of thing. And it's a very interesting fact. Now, how do we do this sort of thing? What is actually involved in looking at these things? So we don't need to go into much detail, but there's these things called ribosomes. That typically, they do translation of genes to proteins. So they turn things that can be into things that are. But in particular, the subunit, which is called, you know, it's regularly called 16S. This is actually used to replicate bacteria. So bacteria actually cannot reproduce themselves or make more bacteria without using this thing. And what's interesting that was found a few years ago is that this gene has some areas that are different from others, much like fingerprints, right? Just like everybody has their own fingerprint, so do bacteria in this particular gene. You can tell who it is by just looking at these genes. And you can tell, yep, that's, you know, Amy, that's Jeff, that's whatever, right? You know, that's who they are. This cannot evolve very fast because it's very essential for their replication. So it's a very particular signature. It's actually even a little better than fingerprints because we can also tell who you're related to. It's almost like you could tell who is your cousin and sister and mother from fingerprints, which we can't do that with fingerprints, but we can do that with this, which is really, really helpful. So we know not only who you are or your name, we also get to know who you're sort of like. So we can try to start to guess what kind of microbe you are, right? It's an extremely useful tool. And so when I joined Mayo, one of the things that we, you know, started studying with some, you know, commitment from clinicians here was endometrial cancer. And some of you may have never heard of this. But this is the uterus, right? And then you have the endometrial lining here. And it's actually number 9 in terms of you know, how many people have this cancer. So this is a cancer that impacts, you know, about your mother, grandmother age type of women. And it can actually you know, be very deadly, and it can be more harmful actually than skin cancer even. So really impacts a lot of people. So the question was, you know, the clinicians here were thinking there's some kind of microbial component to this potentially. Can you look into this and see if, in fact, there is? So Andrea Mariani was the clinician that kind of convinced me of this. And you can see here, you know, this is a study we did. So basically what we did was to look at women who had endometrial cancer and were having a surgery to remove the uterus versus women who had some other problem, but it wasn't cancer, endometrial cancer. So And what you can see here is we basically treated this, put these spatial locations in the uterus to kind of track what kind of microbes they have, right? And you can see by the colors, just by looking at the colors, which represent single microbes, that they are pretty much similar, right, across the uterus spatially, right? And you can see in this plot, which, it's called the principal coordinate analysis, but really what matters here is that things that are, again, close together are alike, things that are far apart are different. And you can see how the patients who are in blue, endometrial cancer, are distinct from the ones that are in red, right? So this is based solely on the microbes they have. So only the microbes that are in the uterus can we tell them apart, and we could, right? And then we looked further to kind of understand what kind of microbes they have. And what you need to understand about this network analysis, is what's called, is that those lines represent how microbes are connected to each other. So who's friends with who, and who's connected to the disease, right? And so the green lines, the thicker those lines are, the more friendly they are with each other. So just see if you're paying attention. Who's the best friends in that image there? The what? Can you say it again? Who said, I could hear, did you say? Yeah. Yeah, who are the best friends on the image? Can you see the names? These up here, right? Yeah, so these guys are best friends. And then the blue lines, you can see the thicker, the more they're correlated with the disease that we're interested in, right? And so you see this particular microbe, right, that is kind of this thicker line really connected to the cancer. And so we started kind of homing in on this one because it's particularly associated with the disease. And so, we've been doing this and looking into these, what turns out to be 17 microbes associated with the disease, and you can see how, as the disease progresses, they get more and more of them as the color gets darker here. We can also do kind of tests just with a swab, actually, on these patients and know that basically if they have this particular microbe, which is Performonas as it's called, if they have it, basically 1 out of 15 women, is gonna have this cancer, and so that's something we're utilizing to identify women early so that we can treat the disease earlier. Where are we trying to do this? Again, we like to travel, remember? So we are particularly interested in studying patients that tend to suffer the most from these conditions. That is particularly Black patients tend to have more aggressive developments of the disease and be diagnosed later. And so we are partnering with Advocate Healthcare in Chicago, Morehouse in Atlanta, and Johns Hopkins to kind of study these patients better. We also have a collaboration that actually emerged from the New Zealand government that reached out to us because their native populations, Maori and Pacific women, have the highest incidence of endometrial cancer in the world. Nobody really knows exactly why, but that's part of what we're trying to help with, is to try to understand if it's a microbiome related situation that is developing there. So the other things we're doing is to kind of study, just because something is associated or correlated with something, does that mean it's involved in the disease? Or is it just something that you can use as a marker, right? So what we're trying to study is, what does that mean? Can we stop the cancer or, prevent it? And so we're looking into seeing what the bacteria is doing to the cells. We have seen that it can invade the cells and it can disturb the pathways. This is a fairly big finding. You can see Taylor Crooks was one of my students that is now at the University of Minnesota, but he was so happy when he found this that he exploded a confetti bomb in my office that you can see here. And he was so excited that he printed a photo of himself that is bigger than him, as you can see, in my office, so that I wouldn't forget about him. And I don't know how I could, because I still have holes in my ceiling from the confetti bomb that he exploded. So yeah, if you think science is boring, that's, yeah. Anyways, and Abby has been working with us a lot on these things as well. Going after, what we call acute and chronic exposures. So this is where we put the cells who are normal, and we put the bacteria, and then keep doing this to see what they start doing to the host cells as they start changing. And at this point, we are actually injecting these in mice to see if they actually cause. We're after causation at this point, and we are seeing some tumor formation that we are investigating to see if that's what's actually happening, which would be a pretty big finding, as you can imagine, but we're really studying to see what we can do about it. Along the way, because of our background in engineering and all these things, we are very interested in kind of tools that can be utilized to play. Very interested in play and just innovation and kind of creating new things. Does anybody know what microfluidics is? Anybody seen this word? No. Well, this is actually quite simple. It's just manipulation of fluids is what it is in a very micro way, right? And I'll show you some applications of this. This allows us to basically play with cells, very, very, very small things. There's different kind of tools that you can use. There's continuous microfluidics where you use just pumps to move the fluids in cells. There's droplet where you use kind of water and oil. What happens if you put water and oil together? They don't mix. That's right. They don't mix, so we can create droplets of this. When we have a droplet platform developed, the droplet platform can produce 20,000 of these per second, so a lot. This is high throughput, right? So you can just create a lot of them. Paper microfluidics, you might remember these from COVID tests or something like that. This is kind of a cheap application of this kind of technology where you can just do quick tests. And then digital, this is kind of the newest thing. Digital is something more like a robot or small robots that you can program and press a button and they do things quickly. So good for automation. You can see these videos here that show these are actually the cells, the bacteria that were in space in this case. But we are utilizing lasers to move them around. You can see that we can pull them. And kind of, this is actually a no-contact technology, is with lasers. So we trap the cells with these lasers, and then we can move them around. Sometimes I have students about your age, come over and they play with bacteria in their mouths. Like, you can use your spit and then play with it, and kind of do this sort of thing. But you learn a lot by just understanding how these bacteria, just by observing what they're doing and being able to manipulate them without actually touching them and disturbing their function. So it's been really an interesting thing to do. As you can see here, the size is very small, so it's micro. Right, it's microfluidics, and we put this together with collaboration with MIT and the University of Utah. But this is basically a microscope that has these laser tweezers and the microfluidics system that we connect to these pumps. And here you can see the digital, which is the program. So this is, you press a button and you tell, oh, go left, go right, go up and down, and they start mixing, as you see. So this is great for applications, for maybe, applications for patients, or people who are not scientists or engineers and, don't have to understand kind of all the ins and outs of how to do these things, but can just press a button and get an answer, right? We try to utilize what's called translational applications, right? How can we simplify? This is a very high-tech technology here. How can we simplify these things for easy use, right, by anybody? And that's what we try to do too. This is the drop-up. So this is kind of a merge of two platforms. And you can see here how small it is, again, when we use these droplets to put the cells inside. Lots of them, and study them, put even for delivery with drugs or other cells, we can do those sorts of things. Kevin was my postdoc, he could also do marking to see which cells are alive and dead and that sort of thing, and then he got really scared with this one because it was like, I don't know what's happening. It was like, oh, it's just, cells smile sometimes, they're happy, that's what they do. It was just a funny thing you saw. Again, translational applications that we can have these kinds of technologies that we are developing. This doesn't seem very intuitive, but actually technologies that we use in space can be translated very well for remote applications, because the situation is actually not very different. If you think you're in a space shuttle, you're not going to have access to high-tech technology or a lot of knowledge necessarily at your fingertips. Same thing for populations that are isolated from hospitals. They're also not going to have that access, and so you need things that can work quickly in place. I don't know how many of you know this man, but Denis Mukwege, he's a Nobel Peace Prize winner in the Democratic Republic of Congo. He, for the work he has done with the women in his country, helping them from violence and a lot of things that they've gone through. Here, what we try to develop with him is a test that can be used for detecting the human papillomavirus, which is a causative agent for cervical cancer. And that we could do this in urine, right? And so the challenge was that, when we were talking with this team, we were like, okay, we understand you don't have a lot of resources, but, can we do a PCR, which is a test you can do with a machine you connect to an outlet, right? He said, you do have electricity, right? And he said, sometimes. And so that was kind of when it kind of hit us like, we can't even rely on that. So we have to do something that is cheap, one that can be distributed by mobile campaigns, and it's under $1. So these are kind of the sticks that we formed, where you can have a sample, and then it kind of basically, if it's a positive, it will turn blue, and if it's not positive, it won't turn blue. And so the women know that if they have it, they can go, to the hospital to be treated early. And before things get too advanced, right? And then they can't be treated anymore in DRC at least. So it takes a lot of kind of sometimes imagination from teams to kind of try to figure out how to do these things, even simple things, as is the same problem with vaccines that we have, right? Which is, the problem is actually not in delivering the vaccines, is in keeping them in a cold place that they don't go wrong. So that's part of sometimes the challenges. I also want to tell you that the sky's not the limit, right? I never, some people say, the sky's the limit. Not in astrobiology, not in imagination, right? Don't let anything limit you. This is something we're currently doing, looking into Martian meteorites and looking for signs of life inside them, with, again, a lot of collaborations from the European Space Agency as well. But utilizing some of these technologies that we keep developing for medicine and for astrobiology, and then we keep going back and forth, because actually the translation is really not that difficult. Other projects that we utilize this technology, Answers in Hours for surgical infections. This is something that we try to give answers to the doctors before patients develop symptoms. Is kind of the goal, right? We don't want to wait until somebody develops an infection to try to do something about it. If we can know right at the time of surgery that you're very likely to be developing an infection, we want to do something about it as soon as possible. And so that's why we are developing this platform that is just very fast. So, all right, last thing for us then to chat more. Last thing I want to leave you with. Solutions to problems are not always where you think they ought to be. This was something that I've learned across along my life. Barry Bloomberg, he was the first director for the NASA Astrobiology Institute, and he was also a Nobel laureate for hepatitis B vaccine development, which a lot of you probably have, and might just save your life. And an incredible person that also crossed all these fields. He has been honored in the Library of Congress for really his commitment to research and dialogue between disciplines. I think, a lot of us, and maybe now especially here in school, and people tell you, you have to choose a path, focus, do one thing. I think there's a lot of value to that and a lot of importance to that advice. But also, if you're the kind of person that just likes to do different things, likes to do things in between other things, likes to be the bridge in essence, I would really encourage you to not let it go. I think that's a really important role that is really important to perform, and be able to be those bridges between fields. Sometimes it's where the big findings are. It's right in between because nobody's looking there, right? So fight for that middle, I think, is important. This is a picture where I brought Carl Pilcher, who's the NASA Astrobiology director at the time, and Heidi Nelson was the Department of Surgery chair. And I brought them together, and Carl was, working with astronauts all his life, but he was fascinated with the OR. When he came to Mayo, it was really, it still talks about this experience that he never thought he was going to see the things he saw Mayo do, right? And then took Heidi to NASA, and so she can see what the astronauts do and all those things. It opens your minds just to be in those environments, right? To see how others do things can really change how you do your own work. This is my team right now with the Mayo brothers. We go to them for a lot of advice. They don't talk much, but they listen. They're all young people like yourselves, as you can see, and they have families. Just got a little baby from Abby. It's a fun job, I think. I don't know, I'm biased, but I think it's fun. Whatever it is you choose, it doesn't matter. Just, this is always what I tell them, and I tell everybody, and my own kids as well, is to dream big and dare to fail. I think we are often paralyzed by fear a lot. What if I fail? What if I can't do this right? That's really important to know. A lot of students sometimes, when they come to my office and they want to try something and then it doesn't work and they feel very sad and discouraged. It's great to know what you don't like. It's just as important as what you like. So don't be afraid to try. Try it, see if you like it. Sometimes you're surprised. And if you find that something you thought you're going to like, you don't like, well, now you know. And you focus on other things then. So just don't feel limited. I really want you to just reach for the stars because they're there. I'm here as a living proof that your dreams can come true because mine did. So I don't see why yours can't. So I hope you keep that fire that you have. Don't let that go away. So it's really important. So thank you, and I'll take any questions and comments you might have. We can chat a little bit.
I'm going to... Is that better? Yes. I'm going to try to pass the mic around so that everybody can hear all of the questions. So maybe I'll start. I have a sort of logistical question when it comes to splitting research between medicine and astrobiology. How does that get handled from a funding perspective?
I think Dr. Lazaridis there wants it's challenging. It is very challenging. I mean, this is, I guess, hard to explain to maybe the younger folks here, but it's very challenging because, so as I said, I am in the middle of things. And for those that like that space, it's a lonely space. So you are basically an outsider everywhere, right? And you're always learning. You're always, when I came to Mayo, I spent 3 years just in the ORs and pathology because I didn't have a medical background and I needed to understand what medicine was to begin with and what physicians did with their patients and all that. So it takes a little longer path, I think, to learn those things. But so you need to be in an environment that is accepting of that and understanding that you're going to bring something different in, right? But it's going to be a little more difficult to explain what is it that you do, and for others to see the value of that can take some more time. But it's if you're fascinated by discovery and just being in a space where no one else has been, which is what drives me, I think it's worth the thrill, just the discovery, just finding new things, being where nobody has ever been. To me, that's science. That's the most exciting part of science. So I really enjoy it, even if it's hard. It's not easy. I'm just going to say that it's not easy, but it's fun. So I think it's worth doing. So.
Thanks for coming out. I come from kind of the laboratory research area, and the idea dare to fail. How many projects or ideas have you come to, let's not say fail, but dead ends?
A lot.
And I guess the big thing for me is, were you allowed to fail?
That's right. That's right. Like just like John F. Kennedy said, we don't do it because it's easy. We do it because it's hard, right? Yes, that is something that actually the motto at NASA now is. So it used to be one of the mottos there that failure is not an option. When I joined NASA, that was the motto. Failure is not an option. Now, the last meeting I had with them, which was just a couple weeks ago, they said that in order to find what the kind of advances that we need right now to get to Mars and other things, basically they asked reviewers of proposals to look for projects that they think are going to fail, and to look for people that they think are going to fail, and actually invest in those. Which is crazy, right? Because it's the complete opposite of how you do this typically. But the idea is to say and accept the fact that they will. That's your assumption. It's like, I don't think this is going to work, so I'm going to give you the money so they can prove me right, basically. And then if they prove you wrong, that's huge, because what they are asking is, look at the impact. What would it mean? Look at the impact as the value of it, right? What would it mean if this person is right, if this project works? What would that mean, right? How would that change completely what we're doing, right? Would that be transformative? Because if the answer is yes, it's worth that failure, the risk of that failure, right? But it is very likely to do that. And again, the expectation is that you're going to fail, and that's okay. That's just the expectation. But they're going to give you the benefit of the doubt and give you everything you need to succeed. So let's give it a shot and see what happens. So that it's an absolute mindset flip from how we think about, typically in medicine, which is a lot more conservative environment, things like that, where you want to sure bets because you want to make sure that there's some progress. But transformative progress just requires a complete change of that mindset and being brave. And really accepting failure as part of the process. Just like we tell our students, right? We say, when they start doing science, your experiments are going to fail, all of them. Any time that a student comes to me with the first time they tried something and it worked, I'm like, you did something wrong. There's no way. There's just no way, right? Because it never works. It's not like that. It takes several attempts to make something work, and that's just part of that process, and that's fine. It's just how you get it right. You keep trying. So, yep.
So I guess when trying to find alien life, most people think that aliens are going to show up in spaceships and talk to us in English, but most likely it's going to be something like detecting oxygen or methane or something in the atmosphere of other planets. Is there other newer techniques that they're looking at for detecting that?
Yeah, actually, their study, and actually didn't talk about MEDI, which I've also been involved in, but that's the one that's actively messaging extraterrestrial intelligence, so that is a little more invasive. But there's been some conversations where we actually think, like we've thought about oxygen, right, as a landmark for life. Like any planet that we find any evidence of oxygen is likely to have life because that's how it happened on Earth, right? But from the perspective of what you think about the odds, if we didn't grow with oxygen, if we weren't in this environment, you would think that actually any planet with oxygen would be completely uninhabitable, because it's a terrible idea, right, to fill a planet with oxygen. It's toxic, even to our cells. It causes aging, and it's toxic to our cells in small amounts. And it's highly explosive. So it's like this planet must be about to blow up. So it's interesting how when you start thinking about other elements that could be conducive to life, how the mindset changes. But yeah, there's some spectral analysis that are being used. Methane, as you mentioned, is another one that can be used as a biology indicator or not. There's also some abiotic ways to produce it. But yeah, those have been the main things we've been focused, of course, water, any evidence of water, but there's different sects in astrobiology. You know, some that look for trying to find things like us, and then others try to find things that are likely to produce complex life, and that's complex organics. So something like Titan, which is very different from Earth, but has an ecosystem. It actually has clouds, it has lakes, it has ice, but it's not water, it's oil. But you can have very complex biology developing in that is not like us, but could have a very similar path to biological processes. So there's those two different sects that look for those kinds of different, so more level of complexity of the chemistry. The more complex chemistry you have in a planet, the likelier the chance that you're going to have biology developing. And the more energy you have available, the more likely, regardless of whether that's what happened here or not. That's some of the ways we think about it. Which might scare you more, because if things aren't like us, then are they going to speak English? Probably not. Yeah.
All right, let's thank our speaker again, please.
Thanks.